Assalamualaikum and hello everyone. We are group 6 and we are going to present our finding on the Neolamarckia genus. Our presentation is based on article Sequencing of the Mitochondrial Genome of Neolamarckia. Macrophylla uncovers divergent structure in genus Neolamarckia. Before we begin, I would like to introduce our team members. Gan Zijian, Siti Sarah Sofia, Kasih Dia Emilia and Tai Ying Jin. Let's dive into this article together. To start, let's look at the taxonomy and classification of this genus. Neolamarckia belongs to the Rubiaceae family, which composed of two primary species, N. cadamba and N. macrophylla, which both of them are monoecious. In terms of geographical distribution, N. cadamba is widely distributed across Asia, including Malaysia, Vietnam, India and southern China. In contrast, N. macrophylla is more restricted and is endemic to eastern Indonesia, especially Sulawesi and the Maluku Islands. Ecologically, these are miraculous, fast-growing pioneer species. They can reach massive heights of 45 meters and are excellent for wetland restoration because of their high water absorption capacity. You can distinguish them by their leaves. N. macrophylla has robust, rose-colored leaves, while N. cadamba has glossy with prominent veins. Beyond their ecological role, these trees have significant industrial and medicinal value. In the timber industry, they are highly prized with their straight trunk, long wood, and low lignin, making them suitable for plywood, paper pulp, and engineered wood. Pharmacologically, they have been used traditionally to treat fever, inflammation, and infection. Modern research shows they contain bioactive compounds with anti-cancer, antioxidant, and antimicrobial properties. Regarding the current research status, most previous studies focus on N. cadamba. However, this study is a major milestone as it marks the first time the mitochondrial genome of N. macrophylla has been sequenced and assembled. Our scientific goal is to compare these mitochondrial genomes to understand evolutionary divergence. This data provides a vital foundation for future biotechnological applications and a deeper understanding of how these two species evolve differently.
Next, I would like to talk about the methodology of this research. So first is sample collection and DNA extraction. Firstly, fresh leaves were collected from an XC2N macrophylla tree. Genomic DNA was extracted using Keijan genomic tips by following standard protocols. The extracted DNA sample was checked by 0.8% agarose electrophoresis to detect DNA samples for degradation and impurity, spectrophotometer and fluorometer to detect the concentration of samples and purity of samples. The next method is library construction and high-throughput sequencing. Both second and third generation sequencings were applied to sequence mitochondrial genome of N. macrophylla. For paired biosequencing, library preparation was performed according to the manufacturer's protocol of SMRT Bell. Fragments greater than 10 kb were size selected using blue piping, and following quality control assessment, qualified libraries were sequenced on the paired bio review system, and the subreads generated were processed by software. For Illumina sequencing, a double-ended sequencing library with an inserted fragment size of 350 base pair was constructed according to the standard process. After completion of library construction, qPCR method and Agilent 2100 bioanalyzer were used to control quality. Qualified DNA libraries were sequenced using the Illumina high-throughput sequencing platform with pair N150 sequencing strategy, and the raw sequencing data were filtered with FastP to remove low-quality reads and adapters. Next, for genome assembly, the paired biosequencing data were assembled into contigs using Canoe. Blast and search against the NCBI NT database was conducted to identify mitochondrial genome sequence from whole genome assembly. Bowtie tool was applied to align sequencing data obtained from Illumina to mitochondrial genome, and the resulting alignments were processed using SAM tools. Sequencing depth at each mitochondrial genomic position was calculated using the depth function in SAMTools. And the next method, phylogenetic and collinearity analysis, to investigate the evolutionary relationships between N macrophylla and its close relatives. A phylogenetic analysis using single copy homologous genes was performed. Four, three or above species in the Rubiaceae family were selected. Comparative genomic analyses were performed through pairwise blast and alignments. Genomic regions with the homologous rate less than 95% were filtered out, and next Circus software was used to generate high-resolution synteny maps. The collinearity relationship was done by comparing the mitochondrial genome using BLASTn, which identify homologous genes and conserved regions, and it was visualized using multiple synteny plot function of TV tools.
To begin, for the results, we assembled the Neolamarckia macrophylla mitochondrial genome into a single circular molecule of 289,793 base pairs. With 100% coverage and 97.24 times depth, we identified 58 genes including 36 protein coding, 19 tRNA and 3 rRNA genes. The genome is notably AT-biased at 55.03% and contains fragmented genes like NET1 and COX2. Our analysis showed that ETG is the primary start codon, while TAA is the preferred stop codon. We identified 128 SSR sites dominated by 80 mononucleotides. Additionally, we found 69 long repeats, which are critical because this element drive the structural recombination and genomic complexity seen in the genus. The 19 tRNA genes all show a canonical cloverleaf structure, while the rRNA genes are highly conserved, specifically RRN26, which has less than 0.5% divergence from N cadamda. We also predicted 486 C2U RNA editing sites, with the NET4 gene being the most heavily edited at 39 sites. We observed significant rearrangement while metabolic clusters are conserved. The CCMC 5srRNA fragment is rearranged between species, several fragments are completely inverted, and the 18S and 26S rRNA distribution change from tandem to space, illustrating high structural fluidity. Phylogenetically, N macrophylla and N cadamda are sister clades that diverged approximately 6.75 million years ago. Synteny analysis revealed that 39.13% of the mitogenome is homologous to the nuclear genome, compared to only 1.2% for the chloroplast, suggesting significant nuclear to mitochondrial DNA transfer. Finally, we examine selection pressures. All protein coding genes are under purifying selection, meaning 2.5% to 99.9% of deleterious mutations are eliminated. Genes for ATP metabolism and the respiratory chain like ATP1 and COX1 show very strong purifying selection, while others like RPL2 are nearly neutral, indicating a high level of functional conservation across the genome. Our discussion centers on the 1.4 times size difference between these species. While N cadamda is larger and bipartite, N macrophylla has a smaller single circular chromosome. This divergence is driven by the expansion of non-coding regions and repetitive sequences in N cadamda, which facilitate more complex genomic conformations. The strong purifying selection we observe confirms that core mitochondrial functions are strictly maintained. However, the loss of key genes like COX2 and CCMC in N macrophylla may reduce its resilience to environmental stress. This likely contributes to its restricted distribution in Eastern and Indonesia compared to the widespread N cadamda. In conclusion, this study provides the first complete mitogenome for N macrophylla, filling a major gap in Rubiaceae genomics. These results are essential for future wood industry applications, conservation management, and understanding how mitochondrial evolution shapes the adaptation of these miraculous trees.
So let's move to the comments part. So on the strength side, though, this is a generally important paper, so it fills a real genomic gap for N macrophylla, and the dual platform sequencing approach is methodological solid. So the combinations of phylogenomics, synteny, and also Ka over Ks analysis and RNA editing prediction in one study is comprehensive. That say the limitations are worth flagging. So the entire study only comes from one specimen, so we can't actually speak to population level variation at all. And the 486 RNA editing sites are computationally predicted, so not experimentally validated. And there's no transcriptomic data, so we don't know how these genes actually behave under stress. And the obvious next step is the population's genomics across the N macrophylla range in Sulawesi and Maluku, and the RNA sequence to validate the editing sites, and stress transcriptomics to see how the two species differ in mitochondrial gene expressions. And the SSR markers identified here are also immediately usable for conservation genetics, which matter given this species is threatened in Indonesia. So in conclusion, this study delivered the first complete mitochondrial genomes of N macrophylla. And the headline finding is that two closely related species diverged only around 6.7 million years ago and show dramatically different mitochondrial genome size and structures, despite their highly conserved protein sequence under strong purifying selections. The structure diverged, but the function didn't. And gene loss in N macrophylla is likely to explain its restricted distribution. And this is a solid genomic foundations, and with population sampling and transcriptomic follow-up, the story will get a lot of richer. So that's all from our group. Thank you.